Hello friends, welcome back to another video tutorial from Shomu's Biology. And in this video lecture, we are going to talk about vaccination types. We're going to talk about different types of vaccines and their use, their characteristics, and also we'll be seeing what are the drawbacks of utilizing different types of vaccines, including the live attenuated vaccines, live vaccines, subunit vaccines and all. So let's begin to talk about it. So the first things first is the overall classification of vaccines. I believe all you know and familiar with the idea of vaccination. And in simple terms, if I say what is vaccine, we can say that it's a suspension of microorganisms that includes antibody production to protect against disease and induces immunity. So in a very basic terms, what we can say is that vaccine is going to induce our immunity. It's going to improve our immunity, because these are nothing but microorganisms. So those microorganisms that can cause infection in us, that can result in diseases in us. So what we are doing is we are ingesting those little dosage of those microorganisms, inactivated form of those microorganisms into our bloodstream, so that our body's immune system is going to become familiar with them and it's going to be influenced by them to produce proper antibody against them. And once our body produces antibody, which is a very specific fighting agent against those antigen or those pathogens, then in future, if that same kind of bacteria or microorganism infect us, then in our body those antibodies are already being prepared, so our body is already ready to fight against them. So we are not going to feel sick about that same microorganism infection again in future. So that's the overall story of vaccines and vaccination. So but how and what kind of microorganisms that we can choose for vaccination, that's always a big question. We now have almost 5-6 different types of vaccination strategies. Not all of them are going to work the same way. They are working in a different way, they have a different principle, and not all of them working for the same disease. For different disease and infection, we have different types of vaccines working better than the other. So let's talk about all these types. We can divide these vaccines into two different types. One is the complete agent. We can take the whole microorganism as a vaccine. I'm just calling it as an agent. So whole agent as a vaccine, or we can take just a subunit or fragment of that microorganism as a vaccine. So with this idea, we have a whole agent vaccine as well as a subunit vaccine. Let me take a color: whole agent and subunit. Now this whole agent vaccine can be further divided into two different types, either inactivated or attenuated. Now there is a difference between this term inactivated and attenuated. Inactivated means killed. That means we just kill that microorganism totally. Okay, we kill it. On the other hand, attenuated one is weakened. That means we are not killing that microorganism, but we are cutting some portions of its pathogenic activity. We are reducing some portions of its pathogenic activity. Now generally microorganisms are killed using formalin solution. We treat them with formalin, and at present no risk of getting the disease at all, because we totally kill that with formalin treatment. And an example of vaccine that is killed is rabies vaccine. We all take this rabies vaccine always, so it's a killed vaccine. So there is no risk of that pathogen to be present in our body again. While attenuated vaccine is when we can use DNA mutations accumulated for a long-term cell culture. So what we do, we continue to change the DNA, modify the DNA with chemical mutagens, is going to change it, and we check that for several rounds and several generations of cell culture. But again, there is a big question mark is always there, because it's not totally killed. That microorganism can reverse to a living pathogen, and that can cause some infection in our body. That's always a possibility for attenuated type of vaccines, while in inactivated vaccine that's not a thing. Okay, so attenuated vaccines, example MMR, mumps, measles, rubella vaccine. In attenuated vaccine, no booster shot is required. Okay, booster shot means, sometimes when we use very tiny amount of vaccination, like very small amount of microorganisms in small dose, we need to take multiple dose in interval, in regular interval. That's known as boosted dose, which we don't need for attenuated vaccine. Okay. Now, what about the subunit vaccine? Subunit vaccine, as per the name, as we know, it's only we take the part of that microorganism, and the part of the microorganism we use adjuvant that increase the effectiveness of the vaccine. Adjuvant means large particles that can be tagged. Adjuvant means this is not a part of the pathogen. We can take large detergent molecules or dead non-pathogenic bacteria. This can be adjuvants, because for generating immune response in our body needs to show a kind of very large molecule. That's why the antigens that needs to work, they are bigger in size. Very small molecules are not going to trigger our immune response much, so that's why we need to add these adjuvants. We can take other inert proteins, or we can take portions of dead pathogen, or we can take detergents as the adjuvant to induce the effectiveness of this subunit vaccine. And the subunit vaccine is safer than attenuated vaccine. No risk of getting the disease, because in attenuated vaccine there is a fair chance of reversal of the disease, which is not present in subunit vaccine. But again, subunit vaccine sometimes need repeated doses. Okay, example of subunit vaccine is hepatitis B vaccine. Okay, so these are the differences. We can define the vaccines in different terms. We can also classify vaccines in many other way, but this is the easiest way that we can classify vaccines. Now, based on this classification, let me share few examples of vaccines with you, and we continue with the live attenuated vaccines, or LAV. Example, tuberculosis, that is BCG vaccine; oral polio vaccine, OPV, which is very common in India especially; measles, rotavirus, yellow fever vaccine. All these vaccines are live attenuated vaccine. Inactivated vaccines, which are killed microorganisms, that we use whole cell pertussis vaccine and inactivated poliovirus IPV vaccine. On the other hand, subunit vaccine, which is simply known as a purified antigen, that means we take the whole organism, we fragmentize it, we take only few portions, few subunits, purify it, and we use it. Acellular pertussis vaccine, Haemophilus influenza type B, Haemophilus influenza type B, or HIB vaccine, and pneumococcal vaccine. There are examples like PCV7, PCV10, and PCV13, as well as hepatitis V vaccines can be used. The Hep V, that's the part of the coat, that can also be used as a purified antigen subunit vaccines. So you can see for hepatitis we have both live attenuated vaccine, as well as we have this kind of subunit vaccines here. And toxoid vaccines are where the toxins that are generally secreted by microorganisms, we take those toxins and we exclude the toxicity part. We call it inactivated toxins or toxoids, and we do the same for toxoids that we do for inactivated vaccines, that we treat that toxins with formaldehyde and with high temperature, and that's going to convert that toxins into toxoid. And we use tetanus toxin, diphtheria toxin, and we convert that tetanus and diphtheria toxin to toxoid, and we can use that for that particular disease, tetanus and diphtheria. So generally tetanus toxins and toxoids are very common. We generally take tetanus shot. It's not going to induce our immunity for a lifelong time. We just need to take that. It's going to valid for only 2021. There's even 1 month, 2 months duration only, because it's only toxins, not the whole body. It's not even the subunits, it's the toxins that is produced by the microorganism. And generally these toxoids are produced from exotoxins secreted by gram positive as well as gram negative bacteria. So that's what we convert into toxoids, and we can treat it, we can use it. Okay. Now the final thing regarding all these toxins, and also let me tell you the drawback of utilizing every single toxins with example. For a summary, you go with live attenuated vaccines, weakened version of living microbe that can't cause disease, but the challenge is that there is a chance of mutation, there is a chance of reversal. The example, measles, mumps, rubella, polio, which is known as Sabin vaccine, yellow fever, all live attenuated vaccines. Second type, inactivated or killed vaccines, microbes killed with chemicals, heat, or radiation. Challenges, although inactivated vaccines are safest, but weaker immune response, and they need boosters, they need a repeated interval of doses. Example of this kind is cholera vaccine, flu, hepatitis A, Japanese encephalitis, plague, polio, and rabies vaccines. Third type, subunit type of vaccines, includes antigens that best stimulate our immune system. Sometimes you also need to use adjuvants to induce. So challenges are that identifying a specific antigen from a whole microorganism takes time and takes a research. So development of these vaccines are expensive and time consuming. And examples: hepatitis B, pertussis, pneumonia caused by Streptococcus pneumoniae. Okay, and toxoid. Toxoid, again, formalin inactivated toxins used as vaccines, used when main cause of illness is a bacterial toxin. So examples like tetanus or botulism, generally caused by only toxins adverse effect. It's not going to be caused by the bacterial presence itself. If the bacteria releases toxin, then only the disease happen. That's why in those diseases only we use toxoids, otherwise we cannot use. That's why the example diphtheria and tetanus. Conjugate vaccine. Conjugate is specialized subunit vaccine where antigens are linked to polysaccharides. As I said, like, antigens sometimes need to be linked with larger protein fragments and polysaccharides. These are most effective for immature immune system or for infants. In this case, the immune system is not well developed, and if you provide the antigen directly, their body is not going to generate an immune response based on that small antigen only. So in this case, they are going to need this conjugate. They need these polysaccharides along with the vaccines. Okay, so that's why we use these conjugates to use it. Examples: influenza, type B, pneumonia caused by pneumonia. Okay, DNA vaccine. We also use the DNA fragment as a vaccine. DNA of important antigens introduced to cell, but its role, its proper use, its challenge, it's still not known because it's in the experimental phase. People try to use influenza and herpes as well as HIV vaccines production with the help of this DNA modification, and generally they are linked with the DNA mutagenesis to induce this antigenic feature, immunogenic feature in our body. Then final is recombinant vector, attenuated virus or bacterium used to introduce microbial DNA to cell. It's just like RDT, recombinant DNA technology based vaccines. It's kind of related to the DNA vaccine, so that's why both are experimental level, and both researchers are working for HIV, rabies and influenza more, because what we are trying to figure out here is that if we are using recombinant DNA plasmids, that means if you find out the gene products that can induce that immune response in our body, and if we just insert that gene product in a vector and transfer the vector inside the cell, then the cell is going to have this response. That's what we can do about this recombinant vector as well as this DNA vaccines. Drawback of different types of vaccines. So that's kind of the end, because we already talked about the drawbacks of the vaccine. Don't need to discuss about that either. We already talked about it. So that's regarding the different types of the vaccines, how they are used, why they are used, and all the examples that are very much famous for each of the vaccine types. So if you like this video on vaccination and types of vaccines, please hit the like button, share this video with your friends, and subscribe to this channel. Click that subscribe button and also click on that notification bell so that you get a response from YouTube whenever I upload a new video, and stay tuned to get more interesting video like this. Thank you.